Epilepsy, a complex brain disease with multiple impacts. This expanded audio summary will explore a scientific review article. Epilepsy is more than a simple seizure disorder. Causal relationships between epilepsy and its comorbidities, published in the Veterinary Journal in 2024, authored by Peek et al. This review investigates the intricate connection between canine epilepsy and its commonly associated conditions. Cognitive impairment (CI), attention deficit hyperactivity disorder, ADHD, like behavior, anxiety, and fear. Due to the limited research available specifically on dogs, the review relies heavily on insights from human and laboratory animal studies. Introduction: Redefining epilepsy. Epilepsy, a chronic brain disorder, is most prominently marked by recurring epileptic seizures. However, it's becoming increasingly clear that the impact of epilepsy extends far beyond these seizures, encompassing a spectrum of neurobiological, cognitive, and social changes. This realization underscores the importance of understanding not just the development of epilepsy, known as epileptogenesis, but also the various comorbidities that often accompany it. This review focuses on unraveling the pathological mechanisms that link epilepsy to these associated conditions. The authors emphasize the need to shift away from the outdated unidirectional view that positions epilepsy as the sole cause of these comorbidities. Instead, they propose a more nuanced understanding, highlighting the concepts of bidirectional connections. Epilepsy might contribute to the development of comorbidities, while these comorbidities could, in turn, influence the course of epilepsy. Shared common pathological processes. Both epilepsy and its comorbidities could arise from a shared underlying cause, representing different facets of a single malfunctioning system. This shift in perspective recognizes the complex interplay between epilepsy and its comorbidities, paving the way for a more comprehensive approach to research and treatment. Study design: synthesizing existing knowledge. It's important to note that this review article does not present a new study with its own methods and results. Instead, it systematically synthesizes existing research from human and animal studies to offer a holistic view of the complex relationship between epilepsy and its associated conditions. This approach allows the authors to draw upon a broader knowledge base and identify patterns and connections that might not be apparent from individual studies. Unraveling the connections: common pathological mechanisms. The review focuses on 4 key areas to explore the potential links between epilepsy and its comorbidities: anatomical relationships, functional relationships, neurotransmission, and inflammation. Let's break down each of these areas in more detail. 1. Anatomical relationship. The limbic system is a central hub. A key insight emerges from examining the anatomical structures involved in both epilepsy and its comorbidities. The limbic system, particularly the amygdala and hippocampus, plays a crucial role in both seizure generation and various brain functions that are often disrupted in these associated conditions. The limbic system. A closer look. The limbic system is a complex network of brain structures deep within the brain. It is responsible for emotional processing. The limbic system is the brain's emotional center, playing a critical role in experiencing and processing emotions like fear, anger, and pleasure. Memory formation. The hippocampus, a key structure within the limbic system, is essential for forming new memories and consolidating short-term memories into long-term ones. Learning and motivation. The limbic system is involved in learning and motivation, associating experiences with rewards or punishments and driving behavior. The amygdala, fear and anxiety. The amygdala, a small almond-shaped structure within the limbic system, is central to the experience and processing of fear. Increased activity in the amygdala can lead to anxiety, and both clinical and preclinical studies have demonstrated that electrical stimulation of the amygdala can trigger both anxiety and seizures. This suggests a potential bidirectional relationship or shared pathology between anxiety and epilepsy. Seizures originating in the amygdala might act as endogenous stimulators, triggering anxiety. Pre-existing anxiety disorders, characterized by excessive firing of neurons in the amygdala, could contribute to the development of epilepsy. The hippocampus, memory, cognition, and seizures. The hippocampus, another crucial component of the limbic system, is intimately involved in memory, cognition, and the experience of fear. It is also frequently identified as a seizure-initiating area, particularly in focal epilepsies like mesial temporal lobe epilepsy (MTLA). Hippocampal sclerosis, a potential common link. Hippocampal sclerosis (HS), characterized by loss of neurons and subsequent structural changes in the hippocampus, is a common feature in MTLA. The causal relationship between HS and epilepsy is still under debate, with evidence supporting both the possibility that HS causes epilepsy and that epilepsy leads to HS. However, HS is independently associated with cognitive impairments, suggesting a couple of possibilities. HS could be a common cause, occurring prior to and contributing to the development of both epilepsy and CI. HS could be a consequence of similar pathological processes that underlie both epilepsy and CI. Hippocampal asymmetry in canine epilepsy. Research has shown that hippocampal asymmetry, potentially indicative of unilateral HS, is observed in dogs with idiopathic epilepsy i.e. Additionally, decreased hippocampal volume, which could suggest bilateral HS, has been observed in dogs diagnosed with canine cognitive dysfunction (CCD). These findings provide further support for the hippocampus as a critical link between epilepsy and CI in dogs. Brain structure beyond the limbic system. While the limbic system stands out as a key player, research also points to alterations in other brain areas in individuals with epilepsy and comorbidities. For instance, a study of children with both ADHD and epilepsy found differences in brain structure compared to healthy children and those with epilepsy alone. These differences included increased volume of frontal lobe gray matter and reduced total brainstem volume in the group with both epilepsy and ADHD. These findings suggest that different pathological mechanisms may be at play in varying combinations of epilepsy and its comorbidities too. Functional relationships, exploring brain networks. Moving beyond individual structures, the review highlights the importance of understanding functional brain networks, which represent the coordinated activity of different brain regions. Techniques like electroencephalography (EEG) and functional magnetic resonance imaging (fMRI) allow researchers to visualize these networks and how they are affected by epilepsy and affective disorders. Challenges in canine studies. Applying fMRI and EEG in dogs presents unique challenges compared to human studies, which has limited the availability of canine-specific data in this area. fMRI challenges. fMRI requires the subject to remain perfectly still during the scan, which can be difficult to achieve in dogs, especially those with epilepsy, anxiety, or behavioral issues that may make them more sensitive to the noisy scanner environment. Training a dog to remain motionless for an fMRI scan can be a lengthy and demanding process. EEG challenges. EEG involves placing electrodes on the scalp to record brain activity. This can be tricky in dogs due to variations in skull and brain size, as well as the presence of large temporal muscles that can interfere with signal quality. While EEG can be performed on ambulatory dogs, sedation is often required to obtain clear and reliable data. Altered functional connectivity and epilepsy. Despite these challenges, studies in humans with epilepsy, particularly TLE, have revealed significant alterations in functional connectivity (FC), the coordinated activity between different brain regions. These changes in FC are often observed in seizure initiation zones. Areas of the brain where seizures originate show altered connectivity patterns. Cognitive and memory networks. Regions involved in cognitive functions like memory also exhibit disrupted connectivity. Furthermore, research has shown that higher seizure frequency can further impact brain activation patterns, suggesting that the severity of epilepsy can influence the extent of functional disruptions. For example, individuals with TLE and high seizure frequency show a more widespread and less focused brain activation pattern when performing working memory tasks compared to those with low seizure frequency or healthy controls. Functional connectivity and mood disorders. Disruptions in specific brain networks, particularly the frontal limbic network, are also linked to mood disorders like depression in individuals with TLE. These findings raise intriguing questions. Does epilepsy directly alter FC, leading to cognitive and mood disorders, or could an unknown factor impair FC, resulting in both epilepsy and its comorbidities? Further research is needed to disentangle these complex relationships and determine the direction of causality. Interictal epileptiform discharges, disrupting brain activity. Interictal epileptiform discharges, IEDs, abnormal brain activity detected on EEG between seizures, can also contribute to the development of comorbidities. These discharges can disrupt the normal development of neuronal circuits, particularly in young children, and have been associated with impaired cognitive performance. IEDs and cognitive impairment. Studies in children with epilepsy have shown that IEDs can impact various cognitive functions, including processing speed. The brain's ability to process information quickly and efficiently is affected. Short-term memory. The capacity to hold and manipulate information in mind for a short period is compromised. Visuomotor integration. Coordinating visual information with motor actions becomes more difficult. These findings suggest that IEDs could serve as a potential biomarker for cognitive impairment in epilepsy, but further research is needed to confirm a definitive bidirectional relationship. Shared pathogenesis, a common thread. The observation that similar brain areas exhibit functional alterations in individuals with cognitive and mood disorders, even without epilepsy, lends further support to the idea of a shared underlying pathogenesis. This suggests that these disruptions in functional connectivity and brain activity are not unique to epilepsy, but may represent a common pathway in the development of various neurological and psychiatric conditions. 3. Neurotransmission, chemical messengers in epilepsy and comorbidities. Neurotransmitters, the chemical messengers that allow neurons to communicate with each other, play a crucial role in both the development of epilepsy and the emergence of its comorbidities. The review focuses on 3 key neurotransmitters: serotonin, 5-HT, gamma-aminobutyric acid GABA, and noradrenaline NA. Serotonin, mood, cognition, and seizure control. Serotonin is a multifaceted neurotransmitter involved in a wide range of brain functions, including mood regulation, cognition, and sleep. It also appears to play a role in epilepsy, with studies showing that drugs that mimic or increase serotonin levels can exert anticonvulsive effects. Reduced serotonin and shared risk. Studies have shown reduced serotonin receptor binding, particularly in the limbic system, in both individuals with TLE and those with affective disorders. This suggests that serotonin dysregulation could be a shared risk factor for both conditions, with decreased serotonin levels potentially contributing to both seizure susceptibility and mood disturbances. GABA, the brain's braking system. GABA is the primary inhibitory neurotransmitter in the central nervous system, acting as the brain's braking system to prevent excessive excitation. Its involvement in epilepsy is well established, with many anti-seizure drugs, ASDs, enhancing GABA activity to reduce neuronal excitability and prevent seizures. GABA and anxiety. Interestingly, these GABA-enhancing ASDs also exhibit anxiolytic effects, suggesting a close connection between GABAergic mechanisms and anxiety. Blocking GABA receptors has been shown to increase anxiety, further supporting the link between GABA and anxiety regulation. This shared involvement of GABA in both seizure control and anxiety points to another potential common pathway in epilepsy and its comorbidities. Noradrenaline, attention, arousal, and seizure susceptibility. Noradrenaline is a neurotransmitter involved in attention, arousal, and the body's stress response. Studies have implicated noradrenaline in both ADHD and epilepsy, suggesting a potential link between these conditions. Noradrenaline deficits and increased seizure risk. Research in rats has shown that noradrenergic deficits increase susceptibility to seizures, while substituting NA can decrease seizure vulnerability. Additionally, reduced noradrenergic-tonin-specific brain pathways has been associated with attention deficits and impulsivity in rats with epilepsy. These findings suggest that noradrenaline dysregulation could contribute to both ADHD-like behavior and increased seizure susceptibility. Interconnected pathways, a complex web of interactions. The pathways of serotonin, GABA, and noradrenaline are closely interconnected, influencing each other and contributing to the complex interplay between epilepsy and its comorbidities. Disruptions in one neurotransmitter system can have cascading effects on others, highlighting the intricate nature of these brain chemical imbalances. For inflammation, a silent contributor. Chronic inflammation, a prolonged state of immune activation, can impact a wide range of brain functions, including those involved in the development and progression of epilepsy. While the exact role of inflammation in epilepsy is still being investigated, research suggests that it can contribute to both the underlying disease process and the emergence of comorbidities. Inflammation and epileptogenesis. Chronic inflammation can affect several brain functions involved in epileptogenesis, including neurotransmission. Inflammation can alter the balance of neurotransmitters, disrupting communication between neurons. Blood-brain barrier integrity. The blood-brain barrier, a protective shield that separates the brain from the circulatory system, can be compromised by inflammation, allowing potentially harmful substances to enter the brain. Neuronal excitability. Inflammation can increase the excitability of neurons, making them more likely to fire uncontrollably and trigger seizures. Inflammation and comorbidities. Neuroinflammation is also linked to mood and cognitive disorders, both independently and in conjunction with epilepsy. For example, studies have found elevated levels of inflammatory markers in individuals with depression, anxiety, and cognitive impairment, regardless of whether they have epilepsy. Inflammation in epilepsy and depression. Research has shown an association between increased inflammatory markers and the severity of depressive symptoms in individuals with epilepsy. This suggests that inflammation may contribute to the development and severity of depression in the context of epilepsy. Anti-inflammatory treatments, a promising avenue. Preclinical studies using anti-inflammatory treatments in animal models of epilepsy have demonstrated encouraging results, with reductions in both seizures and depressive symptoms. This suggests that targeting inflammation could be a promising approach to managing both epilepsy and its comorbidities. Mitochondrial dysfunction, a vicious cycle. Long-term inflammation can also contribute to neuronal dysfunction and cognitive decline through its impact on mitochondria, the powerhouses of cells. Oxidative stress, a state of cellular imbalance caused by inflammation and recurrent seizures, can damage mitochondria, leading to a decline in energy production and cellular function. This mitochondrial dysfunction, in turn, can increase neuronal excitability, making seizures more likely, and further exacerbating mitochondrial damage. This creates a vicious cycle where inflammation, oxidative stress, mitochondrial dysfunction, and seizures feed into each other, contributing to both the progression of epilepsy and the development of comorbidities. Dietary interventions and mitochondrial support. Interestingly, research suggests that dietary interventions can play a role in mitigating mitochondrial dysfunction. Enriching diets with medium-chain triglycerides, a type of fat that is easily metabolized by the body, has shown promise in reducing seizure frequency, cognitive decline, ADHD-like behavior, and anxiety in both dogs with IE and individuals with epilepsy. This finding supports the involvement of mitochondrial dysfunction in these conditions and highlights the potential for dietary strategies to support brain health. Discussion, a paradigm shift in understanding epilepsy. This review emphasizes that epilepsy is a complex brain disease with far-reaching consequences, urging a shift away from the simplistic view that focuses solely on seizures. The authors underscore the need to recognize the intricate interplay between epilepsy and its comorbidities, highlighting the roles of neuroanatomical structures. The limbic system, particularly the hippocampus and amygdala, emerges as a central hub in this interplay, potentially serving as both a source of seizures and a site of vulnerability for cognitive and emotional dysregulation. Functional connectivity. Disruptions in the coordinated activity of brain regions, both within and beyond the limbic system, contribute to both epilepsy and its comorbidities, affecting cognitive processes, mood regulation, and behavioral control. Abnormal neurotransmission. Imbalances in key neurotransmitters like serotonin, GABA, and noradrenaline influence seizure susceptibility, mood, attention, and other brain functions, highlighting the complex web of chemical interactions underlying these conditions. Neuroinflammation. Chronic inflammation, often driven by oxidative stress and mitochondrial dysfunction, emerges as a silent contributor to both epilepsy and its comorbidities, influencing neurotransmission, brain structure, and overall brain health. Embracing the common cause theory. The review advocates for embracing the common cause theory, which proposes that epilepsy and its comorbidities often stem from a shared underlying cause. This theory provides a more comprehensive and integrated framework for understanding these conditions, acknowledging the multifaceted factors that contribute to their development. The challenge of bidirectionality. While the common cause theory offers a valuable perspective, disentangling the bidirectional relationships between epilepsy and its comorbidities, particularly anxiety and depression, remains a significant challenge. It's unlikely that a single factor, such as functional abnormalities or inflammation, can fully explain the bidirectional influences observed. Instead, a complex interplay of multiple factors, interacting dynamically over time, likely contributes to the emergence and progression of both the seizure disorder and the associated mental health conditions. Conclusion: a call for continued research and holistic care. Understanding the complex interplay of these factors is crucial for developing effective treatments that address not only seizures, but also the cognitive, emotional, and behavioral challenges that individuals with epilepsy often face. The review calls for continued research to unravel the intricate connections between epilepsy and its comorbidities, particularly in canine IE, where knowledge is still emerging. This research should focus on elucidating the mechanisms underlying bidirectional relationships, identifying the specific pathways through which epilepsy and its comorbidities influence each other can pave the way for targeted interventions, developing better diagnostic tools. More sensitive and specific tools are needed to identify individuals at risk for developing comorbidities and to monitor their progression. Exploring novel treatment approaches, targeting inflammation, mitochondrial dysfunction, and neurotransmitter imbalances, along with dietary interventions, could offer promising avenues for improving both seizure control and overall well-being. This holistic approach to care, recognizing the multifaceted nature of epilepsy, can help individuals live fuller and more fulfilling lives. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.